Good morning, everyone. Myself, Payal, studying in second MSc, Department of Food Science and Nutrition, Uttar College, Mysuru. Today, I am going to discuss a few aspects about the optimization of fermentation time and organoleptic evaluation of pearl millet dosa. Let us know what is fermentation. Fermentation is a process of conversion of carbohydrates to alcohol and carbon dioxide using yeast, bacteria, or combination of both. It takes place in an anaerobic condition. Fermentation is an ancient technique of preserving the food, which is still used in foods like wine, cheese, shrikhand, yogurt, etc. Some of the famous traditional fermented breakfast food includes idli, dosa, dokla, etc. Let us know what are the importance of microbial fermentation. Fermentation improves the digestibility and increases the nutritive value of the millet. Millets naturally ferment from the wild yeast culture. It helps in increasing the acidity and also decrease the pH of the substrate, thereby inhibiting many pathogens. It also enhances the sensory characteristics such as flavor, texture, and taste of the food, and also improve the functionality quality of the food products. It also helps in reduction of cooking time of the food. Dosa. Dosa is a traditionally fermented breakfast food which is prepared using cereal pulses under anaerobic condition. It is originated in South India, which is made by soaking the rice, black gram dal, fenugreek seeds, and which is ground into a thick batter and left for fermentation over a night. Let us know what are the importance of replacing the pearl millet with the traditional dosa. Penicitum glycum is one of the top 5 millet, which is also known as a pearl millet, which is rich in dietary fiber and low glycemic index, helps in reduction of risk of type 2 diabetes. Fermentation of pearl millet increases the nutritive value and also improves the digestibility. It helps in protection of the gut health, provides all the essential amino acid and antioxidants. Let us know what are the objectives of this study. The main objective of the study is to develop the dosa from the pearl millet and to evaluate the organoleptic acceptability of the developed product and to study the effect of fermentation time on sensory attributes. Let us know what are the materials and methods used in the study. The present study was carried out in the Department of Food Science and Nutrition, Uttar College, Mysuru. The raw materials such as parboiled rice, pearl millet, black gram dal, fenugreek, and salt was produced from the local grocery shop in Mysuru. Let us know method. The methods was the sensory analysis was done by the semi-trained panelist using 9-point hedonic scale method, and the proximate analysis was carried out according to the standard AOAC method. This is the flowchart for the preparation of pearl millet dosa. This table indicates the formulation of the preparation of pearl millet dosa, which includes the difference of ratio in the rice and pearl millet, which include P1, a standard which incorporated 100% of a rice, P2, which incorporated the 20% of pearl millet, P3 includes 40% of a pearl millet, and P4 includes 60% of pearl millet, P5 includes 80% of pearl millet, and P6 includes 100% of a pearl millet. This table indicates the sensory evaluation of dosa prepared from the partial replacement of rice with the pearl millet. Among all the formulation, P3, that is nothing but 40% of incorporation of the pearl millet, had a highest sensory score compared to all the formulation. This sensory table indicates the sensory evaluation of pearl millet prepared by the varying the fermentation time. Fermentation time includes 6 hours, 12 hours, 17 hours, and 24 hours. Among all the fermentation times, 6 hours had the dosa prepared from the 6 hours had a highest sensory attributes compared to other fermentation times. This is the nutritive composition of the P3, that is 40% incorporation of pearl millet dosa, which indicated that the protein, fiber, iron, and phosphorus has increased gradually compared to that of the standard dosa. These images indicate the dosa prepared by varying the proportion of rice and pearl millet. The second image indicates the dosa prepared by varying the fermentation time. Coming to the roles of microbes in the traditional fermented products. Microbes involved in the fermentation are generally probiotic in nature and helps in enhancing the human gut health. It possesses the probiotic and therapeutic property. Compared to other food items, fermented foods are rich in protein and minerals. It helps in the production of enzymes and helps in assisting the digestion. It helps in prevention of harmful chemicals during the fermentation process. It also helps in the breakdown of chemical compounds and synthesis of complex vitamins. It helps in reduction of anti-nutrients and increase the bioavailability of vitamins such as niacin, thiamine, folic acid, and riboflavin. It also helps in utilization of iron in the body. It also decreases the preempting diseases such as diarrhea and salmonellosis. The summary of the study. Microbes involved in the fermentation are generally probiotic in nature and helps in enhancing the gut health. It also helps in the increasing the nutritive value of the food such as protein and dietary fiber. It also increases the availability of all the essential amino acids and antioxidants to the body. Among all the formulation, dosa prepared using 40% replacement of a pearl millet had a highest sensory scores compared to other formulation. Dosa prepared with the 6 hours fermentation had a highest sensory scores in the sensory attributes. The proximate analysis of a nutrients in a selected variation of pearl millet dosa had significantly increased the level of protein, dietary fiber, iron, phosphorus, and lower the glycemic index. These are my references. Thank you.